I actually, when did we start, Dave? Probably 10 years ago, I don't know, but I remember that was where I first actually met Eric. And we've kept in touch a lot, and a lot of the people here I know because we're all sort of interested in this kind of thing about understanding more about yourself and improving yourself through self-tracking. And so when Eric told me a few months ago that he was doing this, I, of course, had to find out more and try it myself. And so I'm going to have a slightly different take on how I did things. Those of you who've been following me and some of my presentations I've done with Quantified Self, and maybe you follow me on Twitter and you've seen some of the things I'm doing, I've been very interested in the microbiome for the last couple of years. Because there's a lot of associations that are known between what goes on with all the microbes that are in and on you and the reaction that your body has to them and the food that you eat. And in particular, I was really intrigued by this pretty highly cited paper that was done a few years ago that showed a big difference between the way that some people respond to things like white bread and whole wheat bread. It turns out that a lot of the studies that you read about anything related to nutrition, the conclusions they give are always based on averages and medians, and like the thing is that you're not an average, you're you. And so what you really care about is how does your body react to these things. And this paper presented a way that these scientists thought would let you be able to predict how your body would respond based on the type of microbiome you have. And I know from previous conversations, Gustavo has looked into this a fair bit and has some opinions about this, but I wanted to test it for myself. And you can buy now, so you can get a biome test kit, and those of you who have been talking to me in the past know that I've done now over 600 of the biome test kits of measuring myself pretty much every day since about 2014. My skin, my gut, my nose, my mouth, a whole bunch of things I've been measuring. And I learned a lot with that, but I was really curious to know about these other two companies, Day 2, which is made by the scientists who did that report that came out a few years ago, and another, actually a local company, Biome, it was based in Bellevue, that does a similar kind of test. And I tested with both of them, this was last year, and I found out that their advice was a little inconsistent. One of the tests said that I should, the list of things that Day 2 said that I should avoid, that Biome said I should, that I should indulge in, and vice versa. And that just really complicated things, because now I don't know who to believe. But then I talked to Eric, and he said, we can find out for sure with this continuous glucose monitoring thing. And so here's my receipt. I asked my doctor, and he wrote this prescription right away, and it cost me $78. I just went down to, he had the prescription delivered to Bartell's in Victoria, and $78 I got 2 of them, so that's a month's supply. No insurance, nothing, I didn't have anything special, I just walked in and paid cash for it. And what I got in return was this this little device here that Eric just showed us. And but I also have a late model iPhone, and so I have the iPhone version of it now. And in fact, I will test myself because I happen to be wearing one right now. I will tell you what my blood glucose is right now. So I just reach under my arm like this, and it tells me I'm at 82. Now,
is it trending or stable?
It says it's stable, which is what you'd expect because I haven't eaten in a little bit. Now this is where I'm really hoping that there are some people in this audience who know a lot more about glucose and about diet than I do. Because as I said, I'm just an amateur here, but I will show you in a second a lot of the tests that I did in a previous round of testing when I first tried this last month. But I'm a little bit concerned right now that my glucose is really low. And I don't know if that's good or bad, or if there's something I should do about it. So
How do you feel about it?
I'm a little bit hungry.
You look great.
Low 80s is normal for what it's worth.
I'm afraid that I might have put the device on incorrectly, and that's the only thing I'm talking about, because it's a little bit lower than it was last time.
That's a good place to be.
All right. Well, then you made me feel better. Okay. Yeah, go ahead, Alex.
Just in terms of the inconsistencies between the tests, I mean, did you notice anything else inconsistent about, do they give you a reported breakdown in different types of,
Oh, you're saying the microbiome tests?
Yeah.
Yes. There's a lot, they give you a lot of data, and I analyze it all over the place.
Right.
It's just complicated. And, my my general conclusion is that I think that the microbiome testing is really interesting, and I've learned a lot, and there are a lot of things you can learn a lot, but it's not just as simple as looking at a simple table like that. I think there's a lot more going on.
Yeah. Well, what was this, I guess, like, do you know Phase Genomics in town?
Phase two, no, I don't.
Yeah, so it's a genome assembly company, basically, Jay Shinjuri's lab and another person. I know the CEO very well, but basically, I mean, he basically compares what they're doing, because they do microbiome analysis, to like whole genome sequencing versus like genotyping in the 23andMe SNP kit, and that's kind of what the UBIOMs and biomes do.
Yeah, that's right.
I'm just curious in terms of.
So remember, day two is doing a full metagenomic sequencing.
Yeah, but it's not the sequencing necessarily, it's the way that they assemble the genomes and being able to correlate that to specific types. So like this is Ivan's role.
Yeah, no, and that's right, and that's the secret sauce, and even then, I don't think anybody's cracked it. I think it's just more complicated than
Okay, yeah.
Do you take probiotics?
No, I don't, and I can prove to you that they don't work. That's my answer.
Richard, given your long and amazing history of self-testing, what's your intuition right now about whether metabolomics via blood glucose, and I'm thinking about dimension reduction for you scientists in here, reducing down to blood glucose as the key feature, right, versus microbiome? Because I have my intuition, you can take it for what you want, but my intuition is that blood is going to tell us a lot more about ourselves than microbiome
I'm not
data.
convinced.
You're not convinced?
Yeah, so.
I love this debate. I'm
Yeah,
totally here for
well,
this debate right now.
let me show you kind of what I've learned as a result of this, and then maybe
Okay.
we can talk some more. I think that's a really interesting question, though, but I think the microbiome is going to win. So Eric just showed what's involved in this. It was completely painless. You just stamp this thing. I just stamped it under my arm. It takes 3 seconds. It's just like putting on a sticker on your arm. There's no pain. So the first thing that I wanted to do is I wanted to see how accurate it is. So I made myself one of these. It's a blueberry kefir smoothie. And I got 2 different brands of blood prick test kits. And I pricked myself, same finger, same blood, same blood drop. And I ran them through both machines, and they came back. So this is an hour after drinking the smoothie. It came back, I don't know how well you can read this, but it says 114 in one and 113 in the other. And then I looked at my Freestyle Libre continuous glucose monitor, and I saw the thing on the left was taken at the same time as those 2 readings there, and it says 144, which is totally off. And at first I was worried, but then I waited another 10 minutes, and it came down to 111, which convinced me that it's pretty accurate. It's just there's more of a delay. And part of that is just because the blood is constantly going through your body, whereas this is measuring something called interstitial fluid, which is just the fluid that's among all your cells. And so there's not that heart rate, the heart's just like constantly pumping stuff back and forth. And so it doesn't get the results of the glucose as quickly as something that's blood-based, but there's a little bit of delay. But
Do you know the sample rate for the CGM? Because I thought that they only do it every couple of minutes anyway.
it's every couple minutes, yeah. So it's it's like probably every 10, 15 minutes on the dot.
15.
So that was,
Is it 15? Is it 15? I don't know if it's 15 minutes on the dot or not. It seems like a little
But it
bit.
might
I
have
thought it was shortly last period.
You can test it as often as you want, so I could test it right now again. I'll tell you that since you asked. Now I'm at 82, so there we go.
One thing maybe worth pointing out is that when you scan it to give you instant reading, it tries to predict your current blood sugar value.
Yeah, it's a complicated equation that they use to be able to, there's something that they're sensing, and it has to do with the temperature, and it has to do with some kind of reaction that glucose molecules have with whatever it is that we've got in the filament.
The reading prediction was often way off, but then if I went and downloaded the historic tapes, there was
Yeah, it
reasonably accurate.
Yeah. So one of the first things I tried was I wanted to see what would happen if I did something that day 2 says is really bad for me. So the day 2 test result, you can see on instant noodles up there, it says it's a 4.8, which means it's something I should avoid. And I decided to go all out, and I made myself 2 packs there and see the ingredient list. It turns out, actually, if you do the math on this, if you have 2 packs of this instant ramen like this, without anything in it, it's about the same number of carbs and calories that you get if you ordered a Big Mac, fries, and a Coke. Who knew? Wow. So
That's basically what, all carbs?
it's all carbs, yeah. It's all very fast.
Thanks for taking one from the team. It's a great test.
So I have a question for people who know more than I do about glucose monitoring. It looked to me like the instant ramen is clearly much worse for my glucose levels than you can see. So this is the donut, the red line there is the donut. However, I think quantity matters a lot too. And so having 2 big packs of the instant ramen probably matters more than just having one big bald little donut. What do people think? Are they comparable or what do you think? Is it all just comes down to carbs, the number of grams of
It
carbs?
comes down to calories.
Calories.
I believe the number you want is glycemic load.
Bingo.
You know, so I looked up the whole thing about glycemic load, and in the book that actually Eric cited, written by the day two scientists, their claim is that glycemic load doesn't have any scientific meaning. That it's just a guidance that somebody came up with based on one study that was done back in the 50s with a few people, and that's what it is. So I don't know if anybody can contradict that.
No, I can't contradict that, but in terms of your question, in basic metabolics, it's, your 3 macros in terms of fat, carbs, and protein. They'll get broken down in different pathways, they'll get broken down at different rates, and at the end of the day, fats count for 9 kilocalories, or 9 calories, or whatever. So I think if I were to run like a perfect experiment as a scientist, I would probably control for macros and amount of macros going in, as well as calories.
In grams.
Yeah, or whatever, whatever units you want, pounds if you want. But yeah, really, I think that was what I was kind of asking about your macros and caloric intake. I mean, pizza is like super calorie dense, like tons of fat, and theoretically fat takes like way longer to digest. So, you have just like this huge caloric burden that's just getting processed over time, that's your blood glucose spikes up slowly. I can totally see that happening versus, maybe like something and that just kind of spikes up and goes back down because your body, breaks down to starch in a single reaction.
Yeah, okay, that makes sense. Take a look at this one. So the next one here is, I decided to try pasta, and day 2 says that basil pesto pasta is really good for me, 9.1. And so sure enough, let's see here, it's the basil pasta is the one in blue, I think. Yes, it's the blue one there, which is, it's within, it's within a healthy range. It's not the best one. The best one actually was, Costco sells this pasta mushroom chicken thing. It's really good, but it's very fatty. And I had a bunch of that, and it barely budged my glucose levels. That's kind of fat. And then I tried the basil pasta, the pesto pasta. It rose, my glucose levels rose, but it didn't get anywhere out of the range. But when I had tomato pasta, it was a similar kind of thing as what Eric showed, which is it spiked, and that was also despite the fact that the pasta was whole wheat pasta. So my takeaway from this is, okay, from now on I'm eating very fatty pesto type pastas and tomato sauce type pasta. So the big thing that, and maybe some of you read something that I wrote last year in the online publication Neo.life about my experience with the testing from day 2 and Biome, where they disagreed on whether I should eat apples or not. And so I decided to test to see, should I eat apples or not? Day 2 says no, it says it's a 4.8 out of a 10 scale, but something I should avoid, whereas Biome says I should enjoy it. And when I tested it, lo and behold, I tried twice, and both times it spiked my glucose, so I think day 2 wins.
So that's quantity 1 apple.
That's right, yeah.
Wow.
Yeah. And I tried to do my tests as much as possible, like under the same kind of circumstances. This was taken sometime like in the middle of the afternoon, after my glucose had stabilized. And,
Hold on, is it a Honeycrisp apple or a Gala apple?
Okay, there you go, yeah.
Fresh apple or aged?
Yeah.
I mean,
Yes.
do you think
There's all these other kinds of differences.
do you think adding fat, like adding peanut butter, would change that?
I think it would, yeah.
And that's a great point. Balancing things, balancing fat versus sugars.
Now, for what it's worth, the Day 2 website lets you add different combinations of food. And if I've added, in fact, the site even gives you suggestions for things you might want to add to an apple to make it be better for you. So, and like Eric, I tried just drinking straight apple juice, and sure enough, you get that big
Wow.
spike.
Jeez.
And then I tried oatmeal. Now, again, the microbiome-based Day 2 test says, you can see up at the top, this is a 4.0. I should avoid oatmeal with water, which is kind of weird because I always thought oatmeal was healthy. And that's where you read everywhere is it's healthy. And so when I tested it, you can see here, I think the dark blue one here no, sorry, let's see, the green one is the oatmeal with water. And these are all, I make them all the same way. This is Quaker oats, the non-instant kind, and I boil it. And in the case of the water one, I just boil it with water and then I ate that straight. And then the next day I tried, the red one is with a little bit of milk in it. And the purple one is with cinnamon. So somebody told me that cinnamon helps moderate glucose levels. Well,
In some people,
maybe it does for some people, not for me.
It also depends on the kind of cinnamon.
Yeah, there you go. So, yeah, or how much cinnamon. Anyway, and then the blue one is when I had it with butter. So if I'm going to eat oatmeal now, I'm going to eat it with fat. So you put butter in it, and when you put butter in it, it's just fine. It's that nice, reasonably steady, kind of light blue version of that. So that was an interesting one for me.
Richard, just given what Eric has shown, did you monitor your fitness or your exercise or
Yeah, I did. Yeah.
mobility?
Yeah, yeah. I'm not gonna. I don't have charts to be able to show that here today. I'll show that. Like that's actually an example of something I'd like to do a lot more research on that one. I'll do the next round of testing. So then I tried potatoes, and this is one where I like potatoes, but people say you should avoid it. I don't know. I should. So that blue line there is me eating a potato. Now, I had read, and there's research that says that if you want to reduce the amount of starch in a potato, you can boil it, and then you cool it, and then you heat it up again. So this is a reheated potato, and it spiked my blood. So I don't know what's going on there. And then I tried it with, let's see here, there's one. The green line is when I had a freshly boiled potato with some lamb, some fatty lamb. And that one is actually a reasonable, it's not as big of a spike. And then the red line is actually not with a potato, it's with a sweet potato, eaten with some beans, just for comparison.
So I know we asked Eric this, but I'm curious with you. Do you feel when your glucose is high? Can you perceive that?
Now that I'm wearing it, yes, I feel like I can understand it. I feel like I can listen to my body a little bit more. I definitely feel a lot more aware when it's low. And that kind of goes without saying, because a lot of us, when you feel hungry, hunger is kind of an indication that your blood glucose is low. But for example, after I have the instant ramen or have something else that spikes it, I feel yucky. And you just feel this kind of thing which I now can associate with high glucose. I don't have the charts here, and I haven't been able to do a systematic analysis on this, so I won't show any more charts about this. But when I was testing myself, I noticed that after one meal, my blood glucose should have spiked because I was eating something that had a lot of starchy and high carbohydrate stuff. This was dinner time, and instead of my glucose going up, it went down. In fact, it went down a lot. And I was thinking, wow, this is pretty cool. Why did it go down? And in fact, to make it go up again, I even ate some ice cream, which is what Day 2 says is healthy for me. It's fatty ice cream. It's good for me. And then I discovered the reason why it went down. I had a glass of wine with dinner, and it turns out that alcohol is really, really bad for glucose levels. And
What type of
do
wine?
people know more about this? Maybe somebody can speak over this, but for me, if I have a glass of wine or a pint of beer, it will make my blood glucose go down. And my understanding from reading up on this is that that's because your body treats alcohol like a toxin. And the same thing that's supposed to be going into getting rid of your, the insulin that's supposed to be secreted to get rid of the high glucose is now cut off and says, you know what, we got to stop this, we got to get rid of this alcohol first. Can somebody explain this better than I can?
Physiologically, alcohol, you guys, and for the PhDs in here, correct me if I'm wrong, okay? But physiologically, alcohol causes vasodilation. So there's something that's going on there. It's the reason for flushing. It's the reason for feeling warmer. A lot is happening in terms of what's happening in the blood vessels when you have enough alcohol on board. Then we can start to talk about alcohol crossing the blood-brain barrier and a whole bunch of other things too. It's an orchestra that just cascades in a way that will blow your minds, blow my minds.
Yeah, does somebody have another explanation? Yeah, go ahead.
It depends on the type of alcohol, so there's a bunch of factors. Like with red wine, I did this while I was in ketosis. It didn't even move my glucose by 1 point to take a glass of red wine. So the glycemic index.
Wait, but your glucose was already low then?
Yeah, it was at like, at that point I was at like 67, because I've been in ketosis for weeks.
Okay.
I've tried it while I was not in ketosis, same deal.
Okay, yeah, 'cause for me, it really caused a lunch.
But you mentioned beer, so that's very different.
Yeah, I was gonna say, I can't speak at all to the science, but I can speak to the studies that have been done. And, take it for what it is, the experience with wine was completely different than with beer. That lots of people reported that either it slowed out a spike, not necessarily that it went down, but that it delayed the spike so it was basically flat. But specifically with wine,
Okay, all right, well, that's.
one maybe
so-
because it's wine, and two maybe because it doesn't have, carbs. That it's basically, wine is something.
Well, alcohol metabolizes as sugar in the body, but it also depends what you're eating it with. And red wine does metabolize differently than other alcohols.
When your liver processes it as, the similar process pathway to fructose, it does not go into the bloodstream as glucose. So it's different.
Yeah, so my takeaway was stay away from alcohol. That's the thing, when I saw that.
Especially if you're diabetic and you're worried about like diet.
Yeah, it really messed up my
too low.
glucose levels in ways that was not predictable from what, all the other experiments I had done. And it stands to reason, and I've read and seen things about how, even if you think that it's not interfering with your sleep, it is. And there's just all kinds of reasons why alcohol is just not a good idea. Yeah.
Just a suggestion, there's a Dr. Morris, who's done quite an extensive broad study on diet. And it kind of flies in the face of the popular notions now. She's an epidemiologist, and it's related to the brain and deterioration in Alzheimer's. Like high fast for long term actually aren't good. There's a lot of things that encompass. So it's interesting research to look at people more, about these specifics of blood sugar, but other things that will play longer term into what your goal is, I guess, for what type of health you're thinking about. Do you want to be in the, padded room and with lots of muscles, or do you want to be thinking clearly?
So what kind of numbers are you talking about? You're not talking about like Framingham Heart Study numbers, are you?
Oh, I'm no scientist. I'm just talking about, she's just done, I heard her interviewed, and it's, but look about the fringe of that, but just some things about the range of that. Just
Sorry to interrupt, if we can get Richard to finish, because I'm afraid we have to.
Are we getting,
this room in
so
10
any,
minutes.
I will, so I'm kind of done here, but I will just say that for the sake of science, I am, if anybody would like to join me afterwards, I will go have a beer to see that it really does work this way. So whoever would like to join me, we can go out afterwards. And meanwhile, I want to do a lot more tests on exercise. That'll be my next big thing that I want to try out, and also understanding a little bit more about the relationship between glucose and sleep. And as well as I'd like to try maybe some nootropics, like I'm looking at CBD maybe or something like that to see what glucose affects that. And if you have any other questions or anything you'd like to follow up on, check me out on Twitter.